Right now, deep inside your body, trillions of microscopic recycling plants are broken. Not slowing down, not underperforming, completely offline, because the modern human body is almost never given a break from digesting food. We graze in the morning, we snack at our desks, we eat on a schedule designed not by our biology, but by an industry that profits every time we open a bag, unwrap a bar, or reach for something between meals. And the consequence of that constant feeding is invisible to the naked eye, but catastrophic at the cellular level. Inside your tissues right now, there is an accumulation building. Malformed proteins that were never cleared, worn-out mitochondria that were never replaced, viral debris that was never dissolved. All of it packing into the spaces between your cells, year after year, doing one thing with absolute consistency, accelerating how fast you age. And the mechanism your body uses to clear all of it, the ancient biological protocol that evolution spent millions of years engineering into every single cell you have, cannot activate while you are digesting food. It requires one thing your modern lifestyle has quietly stolen from you: an uninterrupted window of 0 calories. Clinical data now reveals that when you completely remove food from your system, not for an afternoon, not overnight, but for a full 48 hours, your body executes a precise, hour-by-hour biological cascade, a shift from continuous growth and digestion into something far more powerful, a state of deep molecular survival. And the cellular transformation that unfolds inside that window is so structurally significant that the scientist who first mapped its exact mechanism was awarded the Nobel Prize in Medicine. This is what happens to your cells when you don't eat for 48 hours. To understand what makes the 48-hour threshold so extraordinary, you have to understand what comes before it, because your body does not skip straight to its deepest repair protocol the moment you put down your fork. It works through a precise biological sequence, and every stage must complete before the next can begin. For the first 4 to 8 hours after your last meal, your body is running on the food you just consumed. Your pancreas is producing insulin, shuttling glucose into your cells, keeping your blood sugar stable. This is the fed state, normal, efficient, exactly as designed. But by hour 12, the picture changes. Your digestive system goes quiet. Enzyme production slows to a halt. And for the first time in hours, possibly the first time all day, your circulating insulin levels fall to their lowest baseline. This single event is the biological starting gun for everything that follows. At this exact moment, your liver looks inward. It activates what you can think of as your body's backup generator, a molecule called glycogen, which is simply stored glucose, compressed into vault chambers inside the liver itself. Your body begins breaking it down and releasing that stored energy in a controlled stream to keep your brain running and your blood sugar stable while no food is coming in. For the average human, the liver holds roughly 100 grams of glycogen. Think of it as a backup battery with a fixed capacity, not infinite, never meant to be. As you push through hours 16 to 24, that battery reaches 0. The glycogen vaults are empty. Your liver has nothing left to convert. And here, at the exact point where most people assume the body is failing, something extraordinary happens instead. Your body faces the most important metabolic decision of the entire 48-hour window, and it chooses survival. The metabolic switch flips. Deprived of carbohydrates entirely, your liver pivots to a completely different fuel pathway. It begins breaking down stored body fat and converting it into molecules called ketone bodies, specifically a compound called beta-hydroxybutyrate, or BHB, and releasing them into the bloodstream as your new primary fuel source. Now here is where most people get this wrong. They assume ketones are a compromise, a fallback, the body scraping the bottom of the barrel because it has no other choice. The clinical data says the opposite. Your brain prefers ketones over glucose. The neural efficiency of ketone combustion is measurably cleaner. It produces fewer metabolic byproducts. It generates more usable energy per unit than glucose burning in the same mitochondria. And this is precisely why around the 24-hour mark of a well-executed fast, the majority of people report something unexpected. A wave of unusual mental clarity, a sharpening of focus, a quieting of the cognitive fog that most people have simply accepted as a normal part of their day. Your brain has not gone without fuel. It has upgraded its fuel. The neurons are firing with precision. The neural highways are open and organized, and the brain is running on exactly what it was originally designed to run on: the concentrated, efficient energy of fat metabolism. But the ketone transition, as significant as it is not the primary event of the 48-hour window. It is the precondition for it. Because as your body moves into hours 36 to 48, sustained ketosis triggers a cascade of molecular switches that represent the deepest level of biological optimization the human body is capable of executing outside of pharmaceutical intervention. The first switch, the suppression of a master protein complex called mTOR, mechanistic target of rapamycin. When mTOR is active, your cells are in constant growth and replication mode. When mTOR is suppressed, as it is after 36 hours of complete fasting, it signals to every cell in your body that the external nutrient environment is gone and that survival mode programming must now take over. With mTOR silent, a second sensor activates. It is called AMPK, AMP-activated protein kinase. AMPK is your cellular energy alarm, the ancient molecular trigger your body has used for millions of years to detect a famine and respond to it with intelligence rather than collapse. When AMPK fires, it does not initiate cell death. It initiates autophagy. Autophagy, derived from the Greek words meaning self-eating. And before you hear that word and think of wellness trends and intermittent fasting content, understand what this actually is. The 2016 Nobel Prize in Physiology or Medicine was awarded entirely to Yoshinori Ohsumi for mapping exactly how autophagy works at the molecular level. This is not a biohacking concept. This is peer-reviewed, Nobel-validated cellular biology. Here is the precise sequence of what your cells are doing at the 48-hour mark. Once AMPK activates and mTOR goes silent, your cells register that no incoming nutrients are arriving. They begin scanning their own interior for resources. Specialized membrane structures called phagophores begin forming, built from the cell's own material. The cell is literally constructing its own collection vehicles from itself. These membranes extend, curve, and seal themselves around specific targets with a biological precision that no pharmaceutical has yet been able to replicate. Malformed protein aggregates that have been accumulating for years. Dysfunctional mitochondria that have lost their membrane potential. Viral debris lodged in your tissue that has never been cleared. These sealed collection vehicles are called autophagosomes. They navigate through your cell's interior, identifying damaged material and ignoring healthy organelles entirely. Once fully loaded, they travel toward a separate organelle, the lysosome, which is an acid-filled biological vault whose sole function is to dissolve whatever is delivered to it. When the autophagosome fuses with the lysosome, the cellular waste inside is melted down to its molecular components. The broken amino acids are recycled into pristine building blocks for new cellular construction. The remainder is burned directly for energy, the cell fueling itself through the fast from its own recycled interior. By hour 48, this process has been running at system-wide scale for over 12 hours. The accumulation that built up over months and years of continuous digestion being cleared. The biological rust that drives accelerated aging being dissolved. The cellular architecture of your tissues being rebuilt from the inside. However, how you break the fast is as critical as the fast itself. Because your digestive enzymes have been offline for 2 full days and your insulin system is at its most sensitive, a carbohydrate-heavy refeed will spike your insulin aggressively, shock your metabolism.
Of deep sleep architecture. The precise protocol for how to engineer the restorative stages your cells require after a fast is in the video on your screen right now. The biology is waiting. The protocol is proven. Click on the next video to continue. See you there.